Welcome to our final podcast on the digestive system. In this podcast, although we are only covering the liver, I think you'll appreciate that there's quite a bit of material that still remains. As we look at the liver, we're first going to see the arterial flow of blood that comes into the liver, and we'll follow the venous flow of blood out of the liver. This can be a quite complicated process because also we are producing bile. And bile must also flow out of the liver through the biliary apparatus, and so we'll discuss that. We're going to compare and contrast the cells and the organization of the liver, which is critical to its function. And then we're going to look at the functional lobules of the liver. Some of these you can visualize histologically. Others are simply going to be functional divisions of your histology that we'll see. The classical, the portal, and the hepatic acinus, for instance. We're also going to look at the histology of the gallbladder, where the bile is stored, and then we'll look at some of the extrahepatic ducts that are going to transfer this material from the liver to the gallbladder to the duodenum. So the liver, this is the largest gland in the body. Remember, the skin is the largest organ, and it's going to comprise about 1,500 grams of material. Hepatocytes are the principal parenchyma of this gland. They're going to comprise 75% of the stuff that is the liver. Now, hepatocytes are going to produce both exocrine secretions, which is bile. That's going to be excreted into a space called the bile canaliculus. And we're also going to have some endocrine products that we will talk about as we move throughout this podcast. Now, the lobes of the liver are going to be covered by a peritoneum. And this is going to be covered over Glissen's capsule. This is the dense irregular connective tissue capsule of the liver itself with our peritoneum on top of Glissen's capsule. Now, as we look under or maybe the inferior portion of the liver, we're going to see a hilum-like indentation where the vessels enter and leave the liver. And this is referred to as the porta hepatis. And again, you can think of this mostly like the hilum of the liver. In this illustration, we can see the porta hepatis on this inferior side. We can see the hepatic ducts, bile ducts, common bile duct, portal vein, hepatic artery coming up, and this is going to be that surface. We can also see that we have inferior vena cava. This is going to be esophagus coming to the stomach, illustrated in this. Now, this is the typical histology of the liver, and I just wanted to introduce this to you before we begin. What you're going to see in the liver, you're going to see these plates or columns, maybe think of them as cords of cells, that are going to seem to radiate toward a large open space. This is going to be a central vein. And you're going to see this organization throughout the liver, and that's going to be really the chief identifying characteristic of liver as a tissue. So I just wanted to introduce this as we had just looked at the overall organization of the vessels, the position of the liver and the hepatoports, the gross anatomical look. Now, as we look at the blood supply, we're going to focus on getting the blood in and getting the blood out. And we're not going to identify these per se, but this helps with the organization and the anatomy of the liver. For the arterial supply, hepatic arteries are bringing blood into the liver at the porta hepatis. These are going to become ever smaller and smaller and empty blood into the liver sinusoids. We're also going to have venous blood that's coming in through the hepatic portal vein. This is going to deliver blood to the liver as well. The hepatic portal vein is going to give off portal veins. This delivers blood also to the same sinusoid. So you are going to have mixing of venous and arterial blood here. Now, I already pointed out the central vein. This is what's going to drain the blood from these functional components, the lobules of the liver, and the central vein then is going to drain into ever increasing larger veins, the sublobular vein, the collecting veins, right and left hepatic veins before we enter into the inferior vena cava to leave the liver. Now, I mentioned we're also producing bile. So the biliary flow is going to begin as the hepatocytes, the liver cells themselves, remember this is the parenchyma, as they produce bile, they're going to excrete it into a small space between adjacent hepatocytes. Now, you're going to have tight junctional complexes that are going to seal this passageway, but these little channels between the hepatocytes are referred to as bile canaliculi. Now, these are going to increase in diameter as these coalesce together and fuse. The biocanaliculi are going to fuse into canals of Herring, and these are going to fuse into cholangules. And again, these are just ever increasing size of bile transporting pathways. The cholangules are going to fuse into interlobular bile ducts. Then we're going to get intralobular, lobars, etc. That's why we have the eventually here. Then we're going to get in right and left hepatic ducts that are going to go into the common bile duct. And that's how we're going to get the bile from the liver to the gallbladder and then to the duodenum. So a little bit about the cholangules. It's very difficult to find information on these, and these are going to be extremely difficult to find. What we're going to see is the canaliculi are going to be defined and the periphery of the hepatocytes. But then as we see these biocanaliculi fusing into these channels away from the hepatocytes, we're going to see that these are lined with simple squamous epithelium. And as these get larger and larger, and as we move into our intralobular ducts, these are going to be cuboidal. So we increase in diameter and we increase in the size and the height of the cell. We've done that with our salivary glands. We're doing that with our sweat glands, and we're also doing it here with the transport of bile. Now, for our purposes, we are only going to identify histologically the biocanaliculi or bile ducts that are going to be part of the hepatic triad, and we'll get to look at those in just a few minutes. So focus mostly on the biocanaliculi and what we just simply refer to as bile duct. So here we can see the organization. We have our arterial blood supply that is coming through our dorsal aorta. We have the celiac artery coming to the stomach, coming over also to the liver. Now notice we have purple blood in this particular illustration. That's because coming from the intestinal tract, from the stomach, from the spleen, we have all the components that are draining into the hepatic portal vein that go into the sinusoids of the liver, as does the arterial blood. The arterial blood supplying oxygen. This blood is supplying nutrients and materials from the digestive tract that's going to be processed, metabolized, and stored in the liver. And as this oxygenated, deoxygenated blood mix together and this oxygen is used, it becomes venous blood that enters into the inferior vena cava. Here's our organization of the liver. We see our gross anatomical view of the liver. And now as we look at the histological view, you can see that we break this down into these hexagonal shaped structures. And again, this is somewhat how this is going to look with the central vein in the center, the radiating cords and sinusoids of the hepatocytes throughout. And then we're going to have on the periphery, in various corners of these lobules, we're going to have arterial blood, we're going to have venous blood, and we're going to have a bile duct. And if we look at these a little bit more closely, these three associated together are referred to as a portal triad. You can see how arterial blood is going to come in and spill that arterial blood between and for the hepatocytes. This portal vein is going to spill its blood into the sinusoids that drains to the central vein. And in this case, we have a bile duct at the periphery. This is going to be the only one where the flow of material is backwards. It's toward the periphery. This is going to be our biocanaliculi that's going to get ever larger. We can have cholangules in here, canals of Herring, that drain into this bile duct. But principally, we're just going to see the biocanaliculi. So now I want to talk about the three lobules of the liver. What we saw in the previous slide and what we're illustrating here is what's referred to as the classical lobule, and it deals with the flow of venous blood. And so in our organization, we can see our various hexagonal shaped units, and this is going to be the classical lobule. It's what you see histologically, and you can see our triads at the vertices or at the points of our classical lobule. Venous blood will drain from our portal vein through the sinusoids and empty into the central vein. Now, the second lobule is referred to as a hepatic acinus. And this is going to deal with the red or the arterial flow of blood as it's coming in, because even though you've got blood coming in from the vein, you've got blood coming in that's deoxygenated, is rich in materials, but the oxygen is depleted. We have to have arterial blood to keep these cells going. So in the upper part of our illustration, we can see our red hepatic acidosis. It's going to be diamond shaped. And two of our points are going to be at the hepatic triads, and the other 2 points of our diamond are going to be at our central veins. So what's happening here, and I don't like the fact that they've got them pointing to a bile duct that's here in green, but what's going to be happening is you have arterial blood that's spilling from this hepatic artery, and this is going to be flowing toward the central vein. But the closer you are to the central artery here, the hepatic artery, the richer the content of oxygen. So we're going to break this oxygenated blood down into different zones. And so closest to the hepatic artery, or in this case the arterial, depending on its side, it's going to be referred to as the periportal zone. As we get further away and closer to the central vein, this is the intermediate zone. And then as we get close to the central vein, this is the pericentral zone. Now, this is going to be the lowest in oxygen. And so to best utilize the oxygen that's available, hepatocytes are going to have many more mitochondria present, so that any molecule of oxygen that's obtained, it's going to be used to produce ATP. So pericentral hepatocytes have many more mitochondria than hepatocytes that are present in the periportal zone. So here is somewhat of that illustration. Here we can see a hepatic triad. There will be another triad here. Here's our 2 central veins. And you can see the darker color here. They're trying to illustrate the red, the flow of the arterial blood. So here in zone 1, this is going to be our periportal, because it's around the hepatic triad. This is our intermediate zone, zone 2. And here we have zone 3, the pericentral, and that's going to be closest to the central vein and the most depleted in oxygen. They're trying to show the red color being rich in oxygen, less because we've removed the oxygen, and very little in this zone 3, but still enough to keep the cells going because they're so enriched in mitochondria. Now, the third lobule doesn't deal with blood flow, but it deals with the production of bile and the movement of bile. So in this case, our portal lobule is going to be a triangular shape. And even though we're showing from a central vein toward a bile duct, understand bile's not coming out of the central vein. Bile's produced by the hepatocytes and expressed into the bile canaliculi that then flows toward the periphery toward a bile duct. Now, I've mentioned this a couple of times, and let's just cover this before we go any further. The hepatic triad, these are at the points of our classical lobule. That's where you can really be seen present in the connective tissue. And it's the hepatic arterial supply, which in many cases is going to be an arteriole by definition. The hepatic venules, or vein, and then we will have a bile duct, and we're just going to leave that classification as such, as a bile duct. Here in this illustration, we can see our central vein located on the right. We can see our hepatic vein. It says branch of hepatic vein, so potentially a venule. Here we have our arterial supply, and there we have our bile duct. And you can see with the arrows, bile is flowing toward the periphery. Venous blood and arterial blood is all spilling toward the central vein. Now, histologically, we're going to be looking at an arterial supply of blood, which we see our red blood cells present here. This is going to be our hepatic vein, or venule. This is kind of large to be a venule, so we're going to call this a small vein. And in some cases, you may see more than one component of each. So here's another arterial supply here. But then you're going to see these low cuboidal shaped cells, and this is going to be a portion of our bile duct, likely going to be an interlobular bile duct. And so we're going to have reached this cuboid shape. So when you see a cuboid duct, a vein, and an artery, you know you're looking at an hepatic triad, especially when you see hepatocytes lined up in these columns and rows. Now, the hepatocytes are going to be organized by connective tissue fibers, reticular fibers, much like we've seen in many of our other organs. And they're going to be arranged in stacks or plates. As we cut them in cross-section, they're going to look like cords. And these hepatocytes are going to be adjacent to each other, and they are going to form junctional complexes with neighboring hepatocytes. However, between the cords of hepatocytes, you're going to have spaces. And these are going to be the hepatic sinusoids. Now, these spaces, the hepatic sinusoids, they are going to be lined by type 3 capillary endothelial cells. These are going to be our sinusoidal capillaries. And to say they're fenestrated, that's a little bit of, that's a stretch, because we may have half micron gaps between these cells. These holes may be extremely huge. So that's why we refer to these as sinusoidal capillary endothelial cells. There's no membrane. And when you see these in cross-section, the cells, that just looks like bits and pieces of cells. You're hard pressed to recognize them even as intact cells. In this illustration, we can see our hepatocyte, and here we have adjacent hepatocytes on the left, lower right, and the upper portion. And this little compartment, this enlarged compartment between, this is a biocanalicculus. You're going to have tight junctional complexes on either side of this open space, and that's going to contain the bile in this lateral domain. Here we have another biocanalicculus, another biocanalicculus. And then we have our sinusoids. Here's a red blood cell. And then our capillary endothelial cell, our sinusoidal type, is going to have these huge gaps. And they are going to define the sort of inner boundary of the sinusoid. Yet there's going to be a space between the sinusoidal capillary and the hepatocyte itself. These are going to be filled with microvilli. And again, remember, that increases the surface area. These are going to be for absorption. But this creates a space between the endothelial cell and the hepatocyte, and this is referred to as the space of Dissay. And you can see that term down here, the space of Dissay. Anywhere you have an hepatocyte adjacent to a sinusoid, you're going to have the sinusoidal capillary endothelial cells in the space of Dissay. Now, again, anywhere we see microvilli, we are fairly certain we are talking about absorption. And these microvilli for the liver cells, the hepatocytes, is going to increase this surface area by a factor of 6. Now, we don't necessarily want the hepatocytes coming into contact with the blood. We don't need to irritate the red blood cells. We need to keep them from being jostled around. Certainly platelets need to remain in the blood flow so that they don't start a platelet plug. So that's going to prevent the aggregation, the coagulation of blood. But we have sufficient spaces that particulate matter, as small as that half micron diameter, can still gain access to the hepatocytes. So the liver is a detoxification center. It's also a filtration center. And remember that if the spleen is gone, then the liver takes over the function of removing dead and dying red blood cells. So it's no surprise that there are a lot of macrophages present in the liver tissue, and we call them Kupffer cells. That's the proper name for liver resident macrophages. In this particular light level illustration, we can see our hepatocytes, our plates of cells. Here's our endothelial cells that line our sinusoids. We can see red blood cells in here. And here are some large particulate-filled cells, and these are our Kupffer cells that are guarding and lining and patrolling the sinusoids, removing debris and material from that space. Here's an electron microscopic view of adjacent hepatocytes showing the biocanalicculus, and you will see a few microvilli scattered inside this biocanalicculus. We have tight junctions. That's these dark spots you see on these adjacent membranes of our hepatocytes. Tons of mitochondria. Tons of mitochondria. This may actually be in that pericentral zone where we're not getting a lot of oxygen to use. Here, adjacent to a sinusoid on both sides, we can see this very discontinuous looking capillary endothelium. We can see our space of Dissay, and we can see all these small sections through the microvilli of the hepatocyte reaching into that space of Dissay. Here's another view. This has been stained for Kupffer cells, and I think you can see the abundance of these cells that populate any area of the liver tissue. Now we look at the biocanaliculi again. There's so much to see here, and as I've mentioned before, this is the area on the lateral membranes between adjacent hepatocytes. And these seals are present because of fascia occludens, these tight junctions all along the length of the lateral portion of the hepatocytes, except for that small canal. And that's really what creates it. There's no lining, no endothelium. It's just defined by the area that is not tied together by the fascia occludens in which the bile flows. Now, this is a particular stain, and you can see these dark areas flowing between hepatocytes. These are the biocanaliculi. And remember, this material is flowing from a central vein toward the periphery, toward a bile duct. Here, this is a fairly low-level electron micrographic view, and or you can think of it as almost like a high magnification light level view. And you can see a lot of information here. We can see our red blood cells present in a hepatic sinusoid. Here we have our sinusoidal endothelium, the space of Dissay with the microvilli injecting in. Here are our hepatocytes with tons of mitochondria. Within this, you can see the lateral boundaries of our hepatocytes, several of them, and their biocanaliculi. You can see many of these present here. In a higher magnification view, we can see it even better. The biocanalicus with its own microvilli inside. Here's the fascia adherens junctional complexes. Here's our sinusoidal endothelium, the space of Dissay with the microvilli from the hepatocytes. Now, what does the liver do? Well, it's almost like, what doesn't it do? You can't live without a liver. They do liver transplants. The liver can regenerate, and we'll talk about that. But the liver functions in storage. It can synthesize from amino acids. It can synthesize, in fact, glucose, but we're going to store glucose as glycogen here. And we can also store vitamins and minerals, vitamins A, D, B12, K, but also iron and copper are stored in the liver. So people that eat liver, it's a rich source of vitamins and minerals, absolutely. Now the liver, as I mentioned, does synthesize glucose, and it does it from various amino acids. It can do it from triglycerides as well, but it produces amino acids, proteins, not the least of which the liver produces albumin, angiotensinogen, fibrinogen, prothrombin. So all of these important molecules for clotting cascade, for the osmotic balance of the blood. Angiotensin certainly is going to be used to balance the fluid level of the blood. The liver also synthesizes cholesterol, lipoproteins, the HDLs and the LDLs particularly. And of course, we know it synthesizes bile. Upwards up to a liter of bile can be produced a day by the liver. Now, bile is mostly water, and 80% of liver cholesterol that's synthesized is eliminated here. We don't eliminate it all, but a lot of it. And then we're also going to eliminate in the bile bilirubin glucuronide. Now, this is going to be part of that breakdown process of red blood cells, and we'll expand on that in the next slide. But we're also going to eliminate bile salts. This is going to be 50% of the organic part of the bile we refer to as bile salts. Now, as we mentioned, bilirubin, this is going to be the byproduct of the breakdown of red blood cells, the heme group. And this bilirubin is first going to be processed by macrophages. And so we're going to bring in free bilirubin. Now, it is conjugated with albumin to make it water soluble to transport from, let's say, the spleen to the liver. And when this is taken up by the hepatocytes, they are going to degrade the albumin, and they are going to conjugate bilirubin and form bilirubin glucuronide. Now, this bilirubin glucuronide is going to be able to be placed in the bile, and it's eliminated through the feces. Now, we also have products that are urobilinogen. That's going to be removed and processed in your urinary system. That gives our urine its yellow color. This bilirubin glucuronide is going to be broken down into stercobilin, and that's going to give our feces its brown color. So here's just a diagram of some of the functions of the liver. Here we can see glucose function. We can synthesize glucose, or we can break glucose down from glycogen into glyconeogenolysis, and that's going to release the glucose back into our system. Amino acids can be brought in to undergo protein synthesis for all of those different components we talked about: albumin, fibrinogen, angiotensinogen. And then we can also reabsorb some bile salts from the intestine, the acids. Those are brought in, and you can see we're secreting bile salts, cholesterols, cholic acid into our bile canalicculus. Here we're also secreting that bilirubin glucuronide directly into the bile canalicculus, and here we can see that processing of bile that's happening from the hepatocytes. Now, as I mentioned, you can't live without the liver, and fortunately, the liver has this amazing potential to regenerate. And in rodents, it's been experimentally demonstrated that you can lose 75% of your liver, and it will regrow to approximately its normal size. Now, that's amazing because hepatocytes have a relatively low mitotic activity. The turnover rate is usually about 150 days. But if there's a tissue in your body that has the capacity to regenerate, it most certainly is the liver. Now, the liver produces bile. We secrete bile into the bile canaliculi to transport it to where it will be stored and concentrated, and that is the gallbladder. So don't miss that question on a gross anatomical test. The gallbladder doesn't produce bile, it merely stores it. And it can store up to 70 mils of bile in total. Now, when we look at a section through the gallbladder, it's going to be highly folded again. We saw the folded nature to, say, the esophagus. Well, this is going to be that to the 10th degree. But we're not going to confuse that with our esophagus because, number one, it's going to be smaller in size, and we're going to see simple columnar epithelium lining the gallbladder. We will have some muscularis externa. It's going to be smooth muscle, but it's mostly oblique. We're not going to have that clear circular and longitudinal. It's sort of all put together into this obliquely arranged package of muscularis externa. And then we will have the adventitia that's part of the Glissen's capsule of the liver. It's all sort of tied together intimately in that association. Here you can see the folded mucosa of the gallbladder, and it's very clear that we're not looking at esophagus. We're looking at a fairly average-sized organ here. We're not going to confuse this with anything in female reproductive, although this is not unlike what the ampulla of the fallopian tube might look like. This is much larger. Now, if we look more closely, we can see, even at this low mag, we've got our columnar epithelium in the mucosa. We can see lamina propria. We can see the muscularis, that smooth muscle that's present there. And here's another view of that highly folded, simple columnar type epithelium. I don't think even in this we're going to confuse this with anything in the alimentary canal. All that really can be is the gallbladder. Now, as we look at the ducts again that are transporting this material, we've got the common hepatic ducts. So coming from the right and left lobes of the liver, we have the hepatic ducts that are going to unite together into the common hepatic duct. Now, the cystic duct, this is going to be coming from the gallbladder, and it unites with the common hepatic duct to form the common bile duct. Now, a lot of times when we think of vessels and flow, we think of unidirectional. Well, bile is going to be able to move in two different directions. Now, mostly coming from the liver, the bile is moving from the liver toward the gallbladder or the common bile duct to go to the duodenum. But bile that's in the gallbladder, it gets there by moving into that cystic duct to go to the gallbladder. But when the gallbladder contracts, and we're going to see how that happens, bile moves out of the cystic duct and into the common bile duct. So you can see how it moves back and forth. And actually, bile is going to be stored also in the common bile duct. And when that fills up, the overflow is going to spill into the gallbladder, much like the reservoir of your coolant in your engine for your car. When it gets too full, it spills over into that little plastic reservoir. Now, down at the base by the duodenum, we see the connection of the common bile duct with the main pancreatic duct as that's entering into the duodenum. This is going to be termed the ampulla of Vater. And we're going to have various muscular sphincters that are going to assist in the control and release of material into the duodenum. So here we have our right and left hepatic ducts, common hepatic duct. Here we have the cystic duct, and where that unites together, that's our common bile duct. The common bile duct comes down. Here we have the main pancreatic duct. And this is referred to as the hepatopancreatic ampulla, or this is the ampulla of Vater. Do you see this? That union of the main pancreatic duct and the common bile duct. Before we empty into the duodenum, this raised portion on the inside mucosa of the duodenum, that is the papilla of Vater. Now, it's within this ampulla of Vater that we have four muscular sphincters, and these are the sphincters of Oddi. We have the sphincter calloidicus, we have the sphincter pancreaticus, the sphincter ampullae, and then we have this other one that's the fasciculatus longitudinalis. So, the calloidicus, this is going to control the release of bile into the duodenum, and this is going to be around the terminal portion of the common bile duct. Now, the sphincter pancreaticus, this is going to be around the terminal portion of the pancreatic, that main pancreatic duct, and that's going to control the release of that pancreatic juice and all the enzymes into the duodenum. Now, it also, as you can imagine, it's going to prevent bile from entering into the pancreatic duct and getting to the pancreatic parenchyma as well. Now, in the ampulla of Vater, we have another sphincter, and this is going to prevent any material from entering into the duodenum. So these are gatekeepers that are going to prevent material from flowing. However, the smooth muscle of the fasciculatus longitudinalis, this is going to undergo peristalsis in the ampulla, and this is going to facilitate entry of bile into the duodenum, almost like a pump. And I would be very remiss in talking about the sphincter of Oddi if I didn't include a picture of Oddi. And if you don't know Oddi, this is Garfield's friend, and if that doesn't help you, you'll just have to go YouTube that. So here we have our illustration. We have our common bile duct coming down. Here we have our sphincter calloidicus, which is going to regulate that passage of bile. Here we have the pancreatic sphincter, sphincter pancreaticus. Then here we have the sphincter ampullae, which again is going to prevent that material from passing. And then the fasciculatus, this is going to be mostly around in this ampulla area so that we can, via peristalsis, express material out of the ampulla and into the duodenum. Now, again, the bile duct, the common bile duct, it is going to be a storage place as well. You know, most of our blood is stored in our veins at any one given time. So as we close the three sphincter muscles, the bile backs up, and that's where it's going to spill into the gallbladder for that storage. Now, all of these smooth muscles are going to respond to a hormone that's produced called cholecystokinin. Remember, these are cells, the DNES cells in the duodenum. We're going to have some of these in the stomach as well. And when you eat a fatty meal, what this is going to do is it's going to relax those smooth muscles in the sphincter, and it's going to cause the smooth muscle in the muscularis of the gallbladder to begin to peristaltically contract to express the bile out of the gallbladder, down the common bile duct, and into the duodenum.